Hi, welcome everyone to the workshop on synthetic biology. We at SynBio sort of looking to conduct more of these competitions, more of these sessions. The idea is that we at Ideal, our parent company, do conduct a debate tournament for school student across India. But I particularly very interested in areas which are quite dynamic in nature, and B, which are not really penetrated into say Indian education per se. And obviously there are two fields. One is artificial intelligence, generative AI, and all. But I guess there's a lot of opportunity, a lot of buzz about that. But on the other hand, the biology, especially synthetic biology, is an area which includes like lot of recent development, but more importantly, the talk about it, especially in Indian education system, is quite less. And there are a lot of students which come to us, really understand what it is and how it works. And I know particularly about IGEM from my college days at IIT Delhi that it's a very good competition. Uh, they do competition it for both school and college and students, correct? But at school level, none of the Indian team participate. So this is one of the way to kind of make student curious across India for this competition, and where we kind of partner with IGEM from IIT Madras. They are the pioneer along with Indian institutes, where they kind of participate in this competition for regularly couple of years, and they do very well. And that's why they're gonna take you along the journey of synthetic biology. But essentially, the biggest takeaway is that it's one of the most dynamic field. And just being, other than doctor, there are a lot of things which are happening, and I believe this uh, area per se gonna grow bigger and bigger globally, also in India. And that's why we see it's quite exciting. There are a lot of development, a lot of research, not just in healthcare, but in food, climate, across the spectrum. You will see a lot of applications coming, especially with idea of being synthetic uh, biology applicable and available to everyone. So with that, Shreyas and your team, yeah, you can take over and start. And thanks a lot for being here. Yeah.
Thank you. Thank you for that. And yeah, so I'm Shreyas from IIT Madras, and I will be introducing you guys. I think all of you are from the classes 8th to 12th. So I'll be briefly introducing about what synthetic biology is, and I'll be talking about um, we'll start from the basics, from what you already know. I think you guys already have a brief understanding about what cells are, what chromosomes are, what genes are. From there, we'll move on to something even a little bit more complicated, something that is going on in today's world, something research. I mean, something where you actually, I mean, where research is going on in today's world, and a lot of progress is yet to be made. So let us embark on that journey. And all right. So before starting what exactly synthetic biology is, let's first do the basics. Just a second. Yeah. Um, Before that, an interesting quote by Richard Feynman: "What I cannot create, I do not understand." So if we do not understand, I mean, if we cannot create something of our own, like in the biological domain, then we do not understand it, and hence we'll come to it later on about synthetic biology, about what we can create from life. All right. So we'll first talk about stuff we already know. All right. So I think most of you would be acquainted about what cells are, what a chromosome is, what DNA is about. It's the thing that codes our everything in our body, how we are formed, every single thing that we are made up of is because of this DNA, right? And DNA is very similar to what you see in computer. I mean, what you see in your binary format of your computers and everything else. So every genetic information is being stored in the form of DNA. This is something I think most of us are familiar with. And so, all right. And all of the DNA material is stored in your cells inside, I mean, inside your nucleus. And what I think would be new for the high school students would be that DNA is made up of, I mean, this coding substance that codes the DNA is something called the nucleotides. And hence, I mean, and let me introduce you to something called base pairs. So you guys would, I'm sure that you guys would be familiarized with the binary code of computers that is all mostly just ones and zeros. Similar to that, we have something called the quaternary code for a gene, for our genome, or like for biological structures, or like biological information to be stored, that is called the four bases, that is your A, T, G, and C. We'll not go into the details about what they mean, but let me tell you that they're just the same as 0, 1, 2, and 3. So a different combination of this will lead to a different species overall. Like if you have a vast number of these sequences over like billions of pairs, then you find something called your genome. That is different from, I mean, that is quite different from every single organism on Earth. It's different. I mean, I have a different DNA genome, you have a different DNA genome, and so on. It just goes on like that. It's just that these four pairs form the basis of everything, and something called the quaternary code. And here is the full form for your A, T, G, and C. These are different compounds that are very analogous to your binary code, I would say. Just that there are two extra pairs. That is your A, T, G, and C. So that's how it works. And before that, so I'm pretty sure in your 8th and 9th grade, you'd be learning about how protein is the building block for life. Everything revolves around proteins. Everything that is made is just proteins. Everything in your body. And so your proteins. And so for example, your proteins, they make your bones, they help you lift weight, they provide immunity to your blood, they move very essential molecules around and whatnot. They do a lot of stuff that is very useful for our body. And that's how we live. The whole, our body is just made up of these proteins. And how are these proteins made? These proteins are made, and the major, what makes these proteins are something called amino acids. So that's why they tell you to consume protein-rich diet like meat and milk, everything else, because these proteins are rich in these amino acids. And when you match these amino acids in like different configurations, you get something called a protein sequence. And different foldings of these proteins is what makes different kinds of complex proteins that make your body function. Proteins are much like your Lego blocks that come in association with each other. And with every Lego block that you build, you make an even more complex organism. Let's start from bacteria till human life. Everything is on the basis of proteins. But how are these proteins made? So that's how it works. This is known as the central dogma. This is how everything, life revolves around this one particular slide. This is the most important slide in the entirety of biology. So that is called the central dogma of molecular biology. What does this mean? So every protein that we have talked about until now is made by our body. So even if you consume meat, it's first broken down into amino acids, and that is reassembled back as per our requirements, whatever we need. And how does it do that? It has information. This information we talked about in DNA, the quaternary structure, that is first translated into something called RNA, which we'll come to it. And then it becomes these Lego blocks with specific configurations that become proteins. So DNA has the blueprint, like how an architecture has a blueprint of a building, and an architect has a blueprint of a building. Like that, DNA does the same job, and protein is your final build structure, your final finished structure. And everything in between that is what is done by the RNA. So, and obviously, if you need this blueprint to keep continuing on from generations to generations going on, you need something called replication. That is what is called the DNA replication. DNA replicates itself so that the blueprint just gets continually passed down from one generation of cells to another, even within our own body. And then there is something called RNA. So okay, now we'll come to RNA. So DNA is, as we know it, it's stored in our cells or in our nucleus. It can't be moved around the different parts of the nucleus. Different parts of the cell can only stay within the nucleus because it contains information. If anything changes in the DNA, it means we change everything in our body. Like we won't get the desired result of what we want. Like if for example, if a slight change happens in your blueprint, when you were just made as a zygote, then you might not have eyes, you might not have nose, whatnot. We need the specific structure as how you are right now, right? And that has to be preserved, that has to be kept safe in a locker. That is your nucleus. That's where DNA is stored. So it can't be moved around the nucleus, around the cell, giving out instructions to different parts of the cell to do or to make proteins. So that's why we have something called RNA. RNA is something that moves this, this information to specific sites in our cells that make these proteins. All right. So the messenger from the nucleus to what makes the protein is called the RNA. There are different kinds of RNA, but the major, the most important RNA is called the messenger RNA. That is your mRNA, and that is what delivers the message from the nucleus to your protein making sites for making these Lego bricks so that we can build them and make you or make any individual as a person. So this is what RNA does. And this is very important in synthetic biology. We'll come to it as to why RNA is very much important. So the process of DNA DNA becoming RNA, that is your DNA first needs to become RNA. That is called transcription. And once the RNA moves out of your nucleus to give out instructions, that's called translation, because it's going and giving out instruction to these protein makers to make proteins. That's called translation. So this is how an RNA looks like. DNA is like a double helical structure where everything is compact into two like base pairs that are like alternate to each other, and like complementary to each other. Whereas RNA isn't like that. It has an exposed base pair here, as we see it. Why do we need that? We need that because this is like an active messenger, so it needs to give out information, and it needs to attach to these protein makers to tell that, yeah, there is something called UCAG, CUG, that thing that we see in the RNA backbone. So to give out this information to these protein makers, it needs to have exposed ends. And that's why we have something called an RNA, the messenger RNA, that goes and gives information. All right. So this is the structure of an RNA, and that also has a very similar, that also has a quaternary structure, but instead of the A that we saw earlier, it has something called a U. Why is it called U? Just because of a different chemical composition, nothing else. Otherwise, it's the same thing. It's just the ATGC becomes AUGC. So that's the same thing that happens here. So yeah, so this is so what happens in transcription. Now, as I talked about transcription, what exactly transcription is? Transcription is something that happens, that involves the DNA exchanging or like giving out information to this RNA that we talked about, so that it can go and give information to these protein sites outside the nucleus. So how is that formed? That is through another protein that attaches to the DNA every time RNA is made. That is called the RNA polymerase. Polymerase is something that makes. Polymerase in Greek means that it is making it. So RNA maker. So RNA maker is a protein, or RNA polymerase is a protein that unzips your DNA and passes along it, and cordially gives out cordially gives out information that gets transcribed into your RNA. So RNA is just like your DNA, but instead of having two strands, it has a single strand. That'll just come out. Maybe I'll just use a pointer, I guess. 1 second. All right, I don't know how to use a pointer. Never mind. All right. So if you can see this RNA, it'll just come out from the other side of the RNA other side of the RNA polymerase as RNA. And this is what will go and deliver it to outside your nucleus. So this is again. Go to the next slide. Yeah, as I talked about how RNA polymerase comes out from the other side. There is like your RNA polymerase having unzipping your DNA and making another backbone of these AUGCs or ATGCs that just comes out and moves along. Goes out. Goes and delivers message to these protein makers. This is your RNA, and this is the blue one is your DNA, the red one is your RNA. This is how your RNA is made, and this process is called the transcription. Okay? And all right. So, coming to this is a specific part where your RNA loops around itself, which is not important as much. The major thing that you need to understand here is that RNA polymerase makes RNA from your DNA, and in some cases it needs to stop, and that's why it has these. It can't go unzip through every single part of your gene, so it makes these loops that self-attach each other and stop the transcription. So these are called transposons, just for your information. But yeah, it's not as much as important as transcription. We'll come to it later. Now, coming to translation. All right. So what exactly translations? Now we have established that your RNA goes outside your cell. It acts as a messenger, goes outside your cell, and gives it out. Gives instructions to these protein makers. What exactly are these protein makers? There's something called ribosomes. Ribosomes are also proteins themselves. No, ribosomes are also RNAs themselves that wrap around each other. It's called ribosomal RNA. So yeah, before talking about translation, let me give you this piece of information. There are three kinds of RNAs. One is the messenger RNA that we talked about that goes and delivers information. The other is the ribosomal RNA that makes ribosomes or these protein makers. The third is your transfer RNA. I'll be talking about transfer RNA in a few seconds. So what we have right now established is that your purple line that delivers your messages, and this is your protein maker, that is your the green one, that is your ribosomal RNA that wraps around itself and receives your purple strand to make proteins. And depending upon every three triplet of your RNA, it codes for a specific amino acids. And we talked about the amino acids that you eat in meat, that you consume via meat, milk, etc. So these amino acids, they also have, they have something, they have a code, they have a genetic code associated with themselves. All right. So just like how you translate Hindi to English, English to Kannada or something like that, any other language you can take. Just like that, here here we can see your genetic language, that is your AUGC, GCA, whatever, etc. Is getting translated into this protein language. And how does it do so? How does that happen? It happens through the third RNA called the transfer RNA, or the tRNAs. These tRNAs are like small RNA bodies that have a ribosome attached to itself and has like an exposed triplet base associated with it. Like for example, methionine, like the red thing you see here, that is the tRNA. UAC is associated with the with the amino acid compound, that is methionine. There are different kinds of amino acids. They're like around 20 or 25 different amino acids. And so there are 21 amino acids with like 2 or 3 others that have been found recently. So we'll go with the basic 21 amino acids. The 21 amino acids have, every single one of them has like a different code associated with it, a triplet code associated with it. And depending upon your mRNA, the blueprint that carries the message from your DNA, it gets translated. Just like how you translate English to Hindi, your genetic language gets translated to a protein language. And depending upon this, you have the different assemblies, a different assembly of your protein that acts as a Lego brick. So you're basically making a Lego brick here. Just like how your Lego bricks can be like a straight bunch of just a straight line, or like a curved one, or like a or like glass pieces, or like glass dots. They're like different Lego bricks you have. Just like that, you can have different chains of these amino acids put together to form different proteins. and and every individual protein will act as a Lego brick up later on. Like and as you know it, if you have 21 different kinds, you can have like so many combinations of these amino acids to form n number of proteins. This is the most exciting part about genetic engineering, which my cohort will be discussing to you in a few minutes. All right. So now that we have understood what DNA replication, transcription, and translation mean, does anyone have any doubts regarding this? Anyone here?
Hi, my name is Farhan, and I had a doubt.
Yeah.
During DNA transcription, you mentioned there was trans-protein enzymes. Would those be considered ligase, or does ligase act as a different sort of enzyme?
Could you come again? You weren't audible.
Am I audible now?
Yeah.
So during the mentioning of DNA transcription to RNA,
Huh?
you mentioned that there's the usage of trans-protein enzymes or along those lines. Would ligase be considered one of those enzymes, or is ligase a separate enzyme?
Oh, okay. Ligase is an enzyme that basically sticks your base pairs together. Yeah, so you could consider ligase as one of these transprotein enzymes along with your DNA polymerase that that actually combines different your base pairs together in your RNA. So that is correct.
Thank you so
Second
much.
question. Yeah, go on.
Can you hear me?
Yeah.
Yeah. Um, you had mentioned that, the tRNA, it has the amino acids, they have some sort of association with the genetic information, like in the tRNA. So I just wanted to understand a little bit more. What do you mean by association exactly? Like, how can the genetic information be translated into that amino acid sequence? Could you just tell me a little bit more about that?
Yeah, sure. So this is what happens. Now tRNA is also an RNA, and I've told you before about how DNA gets transcribed into RNA. It also has like a huge sequence. So how tRNA works is that so it coils around itself to expose only three base pairs. Like for example, in this MET, you see stands for methionine. It exposes UAC. So every single tRNA sequence has its own three exposed base pairs. Like for example, UAC, it could even be UAG, or like something like UGA. There's You can have all the 64 combinations. And yeah, so you can have the 64 different combinations of base pairs being exposed, and every single one of these combination is associated with what is attached to one amino acids through charges basically. So there's like a charge interaction that happens between the one end of the tRNA and the amino acid when the tRNA is formed. And according to so the other proteins in the nucleus, they make sure that if UAC is formed, if a tRNA with UAC exposed, if the base pairs of UAC is exposed, then methionine should be the one that is going to connect to this tRNA. So that's how you find these tRNAs being associated with your
Hello? Hello.
yeah.
I'm so sorry. My internet got disconnected. I got disconnected from the meeting. I last left when you were talking about the exposed part of the amino acids.
All right. So I talked about the amino acids being. Since you have three base pairs, that means you can have 64 combinations. So uh,
Mhm,
because 4 cube, that will be 64.
mhm,
So
mhm.
you'll have 64 combinations of these exposed base pairs, and by charge interactions at one end of the tRNA, in your nucleus itself, there is there'll be proteins that search for these tRNA. That, for example, it searches for UAC tRNA being exposed, UAC base pair, exposed UAC, and matches it
Okay.
with methionine. And so that's how it makes sure that every single tRNA gets its respective amino acid.
So, once it attaches onto the amino acids, has the translation been done then, or how does it work?
No, no, no.
Like
So
one.
once it's once it's attached to the tRNA, it
Mhm.
gets carried to this ribosomal site, and depending upon, so this is a very temporary attraction. The MET has a temporary attraction
Okay.
association with the thing, so it just gets
Yeah.
ligated onto the protein sequence that it makes later on. Like,
And then they
yeah,
attach together, like it's like they've attached the amino acids, and then the RNAs. I don't know if I'm right. Correct me if I'm wrong. The RNAs just form their own sequence, and since they're connected to the amino acids, is it like that?
yeah. It's so it's wait.
Okay.
Could you come again? I wasn't. Yeah, I lost track. Sorry.
What I meant was, like, in layman's terms, essentially, is it that the RNA, it's like basically connected to the amino acid. So once they meet at the the ribosomal unit or the site or whatever,
Yeah.
they do they like sort of the RNA clumps together in its own sort of pattern, and since it's attached to the amino acids, they also clump together in that pattern as well. Is it like that?
It's something very similar. So I'll put it in like very simple words. So this RNA, the purple one, doesn't associate itself with the amino acids at all. It's just, and this, it's only, so the only thing that actually uh, touches both your mRNA and uh, your amino acids, it's your tRNA. And
Okay.
so since there's like a particular code that is being, that the mRNA gives. So in the similar fashion, you'll have these tRNAs assembling themselves in the like in the ribosomal sites, uh, and after that there'll be another enzyme called, again, the ligase, that will come and attach. For example, UGG is associated with TRP that's coming next. So MET forms a bond with TRP, and that's how your amino acid chain is made. In the
So
slide,
okay.
yeah,
So when
yeah.
you
So
say, like, amino acid sequences, the tRNA is not there, correct? Like, once they are sequenced, the
yeah,
tRNA doesn't stay, right?
it doesn't
Okay.
stay. It just
All
gets
right.
out.
All right. Thank you so much. Thank you so much.
All right. Any other doubts? Anyone has another doubt? Okay, so I'll move on to the next one. Yeah, so these are the different different codes for your translation. Like every PHE, LE, LEU, all these three-letter words, or these three-letter things you see, those are your different amino acids, and your different codons or your exposed parts are respectively associated to your, this is your tRNA, which will expose UUU, and which will carry your PHE amino acids. Then these are the different kinds of amino acids that you see. This is the table that we have found out. The scientists have found out, I would say. Yeah. And all right. So this is the whole thing. What I've talked about is called the central dogma of molecular biology, and everything, life revolves around this one particular um whatever your tenet. So everything for your DNA being transcribed and then translated to proteins, everything, and your body being only made up of proteins, is what everything revolves around. Life revolves around. So okay, so transcription also mean you can't have transcription go on forever, right? Because your DNA is forever, and then you only want certain genes to be, or certain lengths of the of your DNA to be transcribed. So so there's something called regulation of transcription. So that acts as a start. That signals the start of the transcription from DNA to RNA, and signals the stop of the transcription from DNA to RNA. Or For example, let's take, and there are a lot of genes that are not required 24/7. Now let's take the example of um, your lactobacillus. Your lactobacillus is something that um, yeah that digests your lactose. And when there is no lactose, there is no need for this digestion of lactose to be activated, right? So you only need to activate the digestion of lactose only when there's a presence of lactose. So that's how you have to regulate your genes accordance to that. So for example, you see, that's where this thing called operon concept comes into play. where you have something called a promoter, an operator, the whole gene that makes your protein that digests or does some work. Protein is something that does work, whatever you want it to do. For example, digestion of lactose. And every other gene you see is something that is something that regulates the whole digestion process. So here, yeah. So how does it regulate this? So this is how it regulates it. Now, if you cap the gene, then obviously it's not going to get translated. How do you cap it? Like, for example, this is your gene. If you have some stopper along on top of it, then as the DNA polymerase is trying to pass through it, it can't go beyond a point. So obviously it's not going to sequence out, or it's not going to transcribe this one particular sequence when it's after that stopper. This is what is called a repressor that you see here, the pink color thing. So for example, let's assume there is no lactose. Okay, there's no lactose. That means there's no need for the for your lactose digestion, or your lac-Z, lac-Y, lac-A are genes that make the lactose digestion proteins. Okay, so there's no need for these digestion proteins. So at that time, the repressor goes and binds to your operator gene. But when there is the presence of lactose, then what happens is that this repressor, that this is a repressor, is basically a protein. This protein has more affinity, like chemical affinity towards lactose. So it goes and binds with lactose. So that means this end, you see the lac O, that is free. All right. And now if there is any RNA polymerase that is sliding through this sequence of DNA, it transcribes your lac-Z, lac-Y, lac-A, and because of that, your lactose gets digested. But if there is no lactose, your lac I, that always gets transcribed, and that goes and attaches to your operator gene. So now let's make it simpler, like what I actually meant to say. So now there's always RNA polymerase that slides across this. All right, the slides across this this gene. Now the first thing it slides across is the lac1. So it becomes mRNA, and then it becomes the protein, the repressor protein. Since there is no lactose, it goes and attaches itself to the operator gene. Now what happens? The RNA polymerase is still transcribing slowly. It's going. Then it encounters this one repressor protein. Then it's like, oh, I can't go beyond this. There's like literally a blockade here. There's a boulder here. I can't go beyond this. It stops. So there's no transcription of lac-Z, lac-Y, or lac-A. But when there is lactose, your first lac-I, anyways, like irrespective of the presence of lactose or not, it gets transcribed, becomes this protein. But this protein has more affinity towards lactose. It goes and sticks to lactose. So it means this lac-O is free. Because of that, your now RNA polymerase slides across this and zooms past this and goes beyond lac-O and transcribes your lac-Z, lac-Y, and lac-A, and hence it makes your digestion proteins that go and digest this lactose, because the lactose has been found. That's how it happens. So this is how your regulation of transcription happens. Whenever genes are required, whenever proteins are required, that's when transcription happens. Now, if you're eating food, you don't need digestive juices 24/7. So it's based on it's on a similar principle of that. I mean, as in a multicellular scale, that's exactly not what happens. But if you have to look at it at a unicellular level, this is what happens. Go on, Ahan, you had a doubt.
Yeah, I just this lactose, this protein that we are discussing that is needed to digest lactose, if I'm correct. Um, is it just two questions? First of all, when we say when we talk about like lactose intolerance, is that also genetic, and is this the reason why some people are have lactose intolerance?
Yeah, see, lactose intolerance, there are a lot of genetic reasons, there are a lot of environmental reasons. But yes, as
Mhm.
you said, this is one of, I mean, this could be one of the reasons why you could have lactose intolerance. But in the sense, this particular gene sequence is only for the bacteria. But there are other
Right, right,
genes
right.
that also digest lactose for in our body, that
Right.
may have been
Humans also
mutated.
have enzymes.
Yeah.
Right, right, right. And second follow up to that, if like, can we use tools like, for example, gene editing to um I don't know, reverse this lactose intolerance or undo it if I am not, if that's correct.
Exactly. This is the whole basis of synthetic biology. So it's all a build up to
All
whatever
right.
Skanda is going to discuss. So whatever I've been discussing is I wanted to establish some base so that whatever is going to come next is going to be mind blowing. So he'll be talking about
Okay,
it.
okay. Thank you so much. Thank you for taking my doubt in between. Thank you.
Go on, Devanshi.
Lab P, lab O, what you mentioned.
You are inaudible. I can't hear you. Put it across in the chat. I'll look at it a little bit later.
Am I audible right now?
Nope, I can't hear you at all. Just put it in the chat. Right. All right. So this is the whole thing. Again, this is whatever I just discussed about the repressor and everything. No transcription and transcription regulation. It's the same thing that happens here as well. So yeah, so this is what, and yeah, now regulation now becomes something that we can manipulate. As Ahan said, regulations become something we can manipulate, or even change changing sequence is something we can manipulate. That's where biotech comes into play. That's where we can modify genes. That's where we can like stop regulation, or like over regulate, or like over express certain genes, and manipulate them, exploit them, I would say, up to our will. And that's where biotech comes handy. And he Skanda, my other cohort, will be discussing in detail about how we do that. But these are the major applications. Now, since we have made sense about how um, your we have made sense about how regulate how everything works naturally. What I mean, since we know how it works, that means what I can create, we can create something out of it. And as Feynman said, what we can create, we do not understand it if we can't create it. So this is what biotechnology comes into play, where we can make a lot of advancements, we can cure a lot of illnesses, we can make foods more nutritionally more nutritional we can enhance its nutritions and have a lot of benefits. We can make bioplastics, can have a lot of environmental benefits as well, etc. So with that, I think my god Skanda will continue about what synthetic biology is. Yeah, put it across in chat, Devanshi, whenever you can. The doubt. Skanda, you can continue. I'll answer the doubt via chat. Thanks, guys.
Yeah, am I audible? Yeah, so okay. I think I take it out. Yeah, while defining a gene, there is actually no exact definition for gene. Like it doesn't have to be that long, or it doesn't have to contain certain parts. But something which essentially codes for a protein is called a gene. And yes, those LAC constructs you saw, all those are the names of genes. And and in some cases genes do overlap, but doesn't mean they don't function well. They'll have a good coordinated system for the expression of each protein. I hope I answered your doubt. Yeah, genetic disorders are a wide range. There are a lot of reasons for genetic disorders. There might be mutations, mutations of the gene that is changing in the sequence of base pairs, or there might be deletions, that is like the loss of certain part of the genome. There might be additions, there might be replications. So any change which doesn't result in a functional protein basically will, there are some mutations which still result in a functional protein. So they are called silent mutations. They're called synchronous mutations, and they don't result in genetic disorders. So any mutation or any change in genome which ultimately alters the function of the protein which it should result in will cause a genetic disorder. And those changes can be anything. Yeah, yes, most of the genetic disorders boils down to protein function, because proteins are very essential. They are important for every function, every development, every process that takes place in the body. Okay. There is no. I'm not sure. I'm not sure of any specific reason why each amino acid has like a multiple like multiple codons which code for them. But there are some hypotheses uh, uh, which are just based on evolutionary benefits for multiple codons coding for the same protein, if that's what you asked. Cancer has a genetic facet to it, but it is not purely genetic. It is very partially hereditary. But it is not as simple as it is. So I think I'll continue with my presentation for now, and I'll take doubts in a minute. Yeah, one second. Yeah, sorry. I hope everyone can see my screen. Yeah, am I audible? Yeah, thank you. Continue. So synthetic biology is basically looking at biology through an engineer's eyes. So an engineer always wants to construct a system which is optimal and which optimizes formation of some product. So it's basically designing or redesigning rather a biological system for for human needs or for different purposes and to produce new products. And the basic principle that synthetic biology works on is it's very simple. It's basically uh, in mechanical and electronic systems we have different parts, we have different devices, and we have different systems. So if you compare a genetic system to a car, each part, the parts that you just saw, the promoters, the terminators, are function as discrete parts in a device, which are analogous to screws or pistons or gears in a car. And when you put these together, they form devices which are analogous to engines in a car. And when you put a bunch of devices together, they are analogous to car itself. So it's a fully built machine. Another interesting parallel I would like to draw is to electronic systems, where these each of these parts can be thought of as a resistor or a capacitor or an inductor in the electrical circuit. And when you put a bunch of resistors or terminals or inductors together, you get logic gates, you get other electronic devices like transistors, etc. And so just like that, if you put genetic parts together, you get a genetic device as such. And then when you put different electronic devices together, you get a circuit. And similarly, when you put different genetic devices together, you get a genetic circuit, and which I think is pretty cool. So we'll get to some examples in a bit, which will make this clear. Yeah. So okay. So this is analogous to a device. So let's call this a device. This device has different parts, and it has a promoter. As Shreyas explained, promoter is a region in the DNA which is required for the initiation of transcription. As a promoter, and it has a terminator which stops the transcription. And sorry, one second. Yeah, and this GFP here stands for green fluorescent protein, which is basically a protein which as its name suggests, emits under certain light emits fluorescent green color. And RBS stands for ribosomal binding site, that is essential for translation later on the RNA. So this is a DNA part. And now, what does this device? What is the output of this device? So now, uh, yeah. So the output of this device is essentially the GFP protein. So what happens is through the principles of the central dogma, this transcript, this genetic sequence is transcribed into an RNA, which is later translated into a amino acid sequence or a protein. So there's a promoter site and there's a terminator site and there's a GFP gene in the middle. So ultimately, the output of this device is a protein, which is the green fluorescent protein. So this will give a good foundation for what is to come. And so this is pretty cool. These are something called bio art, or it's called Petri art. This is basically the GFP, the green fluorescent protein I was talking about, is naturally found in a lot of sea creatures. It is not found in bacteria or other mammals. So scientists introduced these fluorescent proteins into bacteria, and because of which the bacteria now emit fluorescent colors. And when the bacteria is streaked in in a definite pattern, they form these works of arts, which I think is pretty good. And so yeah, so these are so so let's say so I think you guys are familiar with NOT gates in electronics. NOT gate is basically when the input is 1, the output is 0, and when an input is 0, the output is 1, which is basically the opposite of the input. And we can so in synthetic biology, this is achieved by using two devices. If you look closely, the first device has a constitutional promoter, which means that it is always expressed. So the first device is always expressed. That means this gene, this LAC-I gene, if you recall, codes for a repressor which represses the LAC promoter. So when this gene is transcribed and translated, the repressor protein represses the LAC promoter on the other device, which inhibits the translation. Sorry, it inhibits the transcription of the other device. So in the absence of lactose or lactose-like material, this repressor represses this promoter, and so this device is off, and the green fluorescent protein is not produced. So when this is introduced in a microorganism like bacteria, the bacteria in the absence of lactose doesn't produce green fluorescent protein simply because the repressor is repressing its promoter. Now, in the presence of lactose, this repressor binds to lactose, and this lac promoter is open for transcription. In which case the green fluorescent protein is expressed. So this first device the product of the first device is basically the repressor protein. So in the presence of repressor protein, the green fluorescent protein is produced. And in the absence of repressor, the green fluorescent protein is produced. So sorry, this is basically an inverter or a NOT gate. So when the first device is on, the second device is off, and when the first device is off, the second device is on. Then moving on to some other sorry, yeah. Sorry, yeah. This construct essentially emulates a toggle switch. A toggle switch in electronics is basically a switch used to switch between two states, state 1 and state 2. And sorry, one second. Yeah, I'm sorry. So yeah, we're talking about a toggle switch, right? So this toggle switch has two devices. That in the first device, as you can see, there are two genes. That is LAC-I and the green fluorescent protein. The second device has two other genes. That is TET-R and red fluorescent protein. And let's say this system is introduced into a bacteria. Now this TET promoter is basically acts very similar to a lag promoter. A lag promoter is positively regulated by lactose, and the TET promoter is positively regulated by tetracycline. And to simply put it, the first device is activated in the presence of tetracycline, which is the compound on the right, and the second device is the activated by lactose, which is the compound on the left. And now let's say you want the bacteria to express only green fluorescent protein. What compound would you introduce into the culture? I think if anyone wants to answer.
Sorry, could you come again?
Did you get what I'm talking about? Like, did you
Yes,
understand
yes,
these devices
kind
in
of,
slide?
right. Could you just
Yeah,
repeat the question you had asked?
yeah. So because this TET promoter is positively regulated by tetracycline and the lag promoter is positively regulated by lactose. If I want to express only the red fluorescent protein on the second device, which compound should I introduce to the medium?
Mm. Um, Sorry, I'm not sure of the answer.
Yeah, it's fine. So now let's say I want red fluorescent protein to be to be expressed in the bacteria. So I need the lag promoter to work. So the lag promoter works only in the presence of lactose, because the lactose binds with the repressor, and it allows for the transcription of the lag promoter. Now, when this lag promoter is transcribed, the TETR, which is TETR codes for a repressor which represses the TET promoter in the absence of tetracycline. And if tetracycline is there, it binds to tetracycline, in which case the TET promoter also transcribes. So on transcription of the second device, the the repressor, which is encoded by the TETR gene, binds to the TET promoter, and it closes this whole device. It doesn't allow transcription of the first device, but the red fluorescent protein is produced. So in the presence of only lactose, only the second device is transcribed, and the red fluorescent protein is produced, which so the bacteria will emit red fluorescent light. And in the presence of only tetracycline, the reverse happens. This lac promoter is closed because there is no lactose, and this tet promoter is open because there is tetracycline, and the green fluorescent protein is expressed, and the bacteria looks green. So the color of your bacteria can be regulated by what compound you're introducing into the culture, which is basically a switch. So like you can switch the colors of the bacteria between red and green through this mechanism. Yeah, before I continue, do you guys have any doubts? Because this is pretty involved. I don't expect you guys to understand everything, but I hope you got the gist of it. I'll take some questions now if you have any.
What are these bonds like you see tet promoter and all?
Sorry, can you repeat?
Yeah, so you see these bonds like OH and all. So what are these?
Don't worry about that. This is just a structure of lactose and tetracycline, the compounds I was talking about. They are just bonds of the lactose chemical. These are the chemical structures of the compounds. So the left
One
one is black. Sorry, yeah.
one question. So when you say like lac promoter, so is lactose necessary for this process we are talking about with the bacteria and the fluorescent protein?
Yeah, for the lac promoter to be active,
Lactose is needed,
the
the sugar.
Yeah,
Okay.
lactose is needed. And why? And it is because did you guys like understand what the lac-I gene does? Lac-I gene basically codes for a repressor protein which interferes with the lac promoter.
Right. Right.
So when the lac-I gene is expressed, the lac promoter uh, is interrupted.
Okay.
But in the presence of lactose, the repressor binds to lactose, in which case the lac promoter is free and the lac promoter can be used.
Oh,
So the lactose is essential for the usage of lac promoter. In the same way, tetracycline is essential for the usage of the TET promoter.
all right. All right. Thank you.
So in the presence of only lactose, only
Uh-huh.
device 2 is expressed, which is red color. In the presence of only tetracycline, only device 1 is expressed, which is green color. So this way you can control the color of your bacteria.
Okay. Okay. So the bacteria, you will see like a complete glow from them, like dots of.
Yes,
Okay. All right.
yes.
Thank you.
Just like the
Thank you
ones in
so.
the bio arts, like yeah,
Right.
no.
Right. Thank you so much.
Yeah, I think I'll continue. Uh, yeah, yeah. These are just so AND gates. AND gates in electronics are gates which basically switched on only if both the inputs are on or both the inputs are one. So basically, when this when so you see three devices here, device one, device two, device three. Now for device three to be active, it say it requires a protein which has two units, and these two units, two subunits of protein are produced by two different devices. So the first two devices should be activated for the third device to be active. That is because the third device requires the two protein subunits produced individually by the first two devices to act together, and basically they act basically like an activator to promote the transcription of the third device. So this is analogous to an AND gate. And similarly, for an OR gate, the third device can accept any one of these protein subunits. So if any one of device one or device two are active, the third device is active. That is how OR gates in electronics work. Uh, yeah, yeah. Before I move on to what is IGEM, I'd just like to say like you guys might find it hard to believe these genetic circuits which look so complex and work on very complicated regulatory ways are actually naturally present in every higher level organism like humans during development, during growth and development of humans. A lot of genetic circuits on a very large scale act together to uh, to to produce different proteins which are essential in the growth and development. And which and these genetic circuits are much more complex. They involve like very high fold genetic devices in them, and their regulations are like scientists are still not able to map out. They're not able to place finger on how exactly are these complicated regulations taken place in the body, which is pretty fascinating to me. Even though I've read this a couple of times, I've explained this to people. I'm enthralled by this every time I talk about this. That is basically what that is a very high level primer to synthetic biology, and these are of course the applications of biotechnology and synthetic biology are almost very overlapping, and yeah, a lot of these but the research in synthetic biology and development is still very fresh, and there are still a lot of developments every day. And okay, coming to what is IGEM. So IGEM is a competition that we at IIT Madras participate in. IGEM stands for International Genetically Engineered Machine. It is a competition which is held annually in Paris, and it is based on synthetic biology, and it has different fields through which you can work on real world problems using synthetic biology, and each project has like different parts to it. It has like a wet lab part, it has a dry lab part, computational part, and human practices part, which involves us interacting with people, spreading the knowledge of biology and synthetic biology in specific. Um so in this competition, teams work on real world problems um, uh, in various fields, like from therapeutics to bioremediation to even foundational development of synthetic biology itself. And it's pretty cool. And every project is very unique and uh, has a very characteristic qualities. And so yeah, this will be all in the presentation. Unfortunately, I could not add any of our past projects to the slides that we prepared. But just to give you a taste of what kind of projects we work on. A couple of years back, our college worked on a project of synthesizing an anti-cancer drug using yeast. So initially, what happened was this drug was harvested from a plant in the Western Ghats. Because of this because of the over harvesting of the plant, because of this, the cancer is a very widespread and is a growing disease. The biodiversity of Western Ghats was threatened because of the endangerment of these species in the Western Ghats. So our college came up with a plan came up with a project using synthetic biology to genetically modify yeast to produce this anti-cancer. So yeah, I think this will be all in this meeting. I'll take any doubts if you guys have.
Just one question. Uh, am I audible?
Yeah, you're
Yeah.
right.
So when we talk about biotech, does biotech also fall under this only? This
Yeah, no,
area of biology?
synthetic biology is a subset of biotech. Biotech is a larger field which involves other other assets to it, like
Technology.
not only
Right.
genetic engineering, it involves biomanufacturing, bioprocess.
Okay. And just
So
one more question. This
yeah,
process that has been going on, the dogma, I believe, has it ever been any different since the onset of humans on this planet, or has this been going on for centuries?
no, this has been not since it has been going for a lot, like think millions of years back.
Eons.
But
Right, right.
it,
That is very fascinating.
yeah,
Yeah.
but it is shown through evolutionary studies that initially this DNA wasn't the genetic material. It was actually RNA, and
Uh-huh.
because RNA is single stranded and prone to a lot of mutations, the organisms evolved to make DNA their genetic material so that it is well conserved. It is a double helix structure, in which the two strands are
Um,
bound to each other, which are less
wow.
prone to mutations. And
Yeah.
even today, a lot of viruses, when COVID and other retroviruses have RNA as a
Right.
genetic material. So the central dogma has exceptions, but in higher organisms, this is pretty much constant in all organisms.
Right. Right. And just one last question. I know I've been asking many questions.
No,
Um,
it's fine.
when we talk about genetic mutations, you mean like how is this change in the sequence stimulated? This sequence in the
Sure.
RNA or DNA, how
So,
is
so
it
what
that
happens is,
stimulated?
so a very common mutation is done during DNA replication. So DNA, so when cells divide, their DNA divide too.
Right.
So during division, that the DNA replication is basically not infallible. It is prone to errors, and those error rates are very small, but accumulation of those over time, and like if the mutations are in important regions of the genome, they lead to variation in the sequence. So like when copying one DNA, like while replicating, uh, so during replication of DNA, one another strand is basically built on a template strand. So while building that, the DNA polymerase, which is the enzyme,
Ah, okay.
prone to prone to making errors. Like instead of these bases are complementary. Like adenine A is complementary to
Right,
T,
right.
and
Yeah.
U is sorry, C is complementary to G. And
All
so while adding nucleotides, this enzyme can make errors, which if you're unlucky can accumulate in essential regions of genome and cause problems. That is one.
got
That
that.
is one source of mutation. There are a lot.
All
There are a lot of other sources. You can look it up if you're interested. It's very
all right, all right. Thank you so much. Thank you so
no
much for
worries.
answering all my doubts. I'm going to log off now. Thank you.